Hi friends, welcome back to my YouTube channel. Today in this video I'm going to explain you the structures of purines and pyrimidines. Before that we will be going to know about the nucleic acid, and then we'll be going deep in the purines and pyrimidines. Okay, so let's start with the nucleic acid. Everyone knows that nucleic acids a majority of two types, right? Nucleic acid, it is majority of two types, and those we know that is DNA and RNA. DNA means deoxynucleic acid, and this is ribonucleic acid. We know that. So if we see the structure of deoxynucleic acid, we'll be having at the second position oxygen is not present in the DNA, while at the second position in the ribose oxygen will be present. That's why that is called as ribose, and it is called as deoxyribose. So all the structures are same, but the difference is only OH and H. Here you can see in this place H2 will come, and at this place OH, CH2OH, and here you will be getting OH. So it is D-ribose, right? So yeah, here at the second position, this is the second position, this is the first carbon, this is the second carbon, the third and fourth, and this fifth, and this is sixth. Now we'll be going to see here first carbon, second carbon, right? So here at H, here OH will come. So this is the difference. At the second position, this is deoxyribose, and this is RNA, okay, ribose. So here deoxy, because of second position, right? So this is DNA and RNA. First time nucleic acid was detected by the Frederick Miescher. He detected the nucleic acid and named it as a nuclein. That time. Okay. After that, there was one reagent called fusion dye. That reagent is used to detect what? It is used to detect the nucleic acid. So what Frederick Miescher, he detected the nucleic acid, and that time he named it as a nuclein, and some other scientists came and they detect the fusion dye as a reagent which detect the nucleic acid. And what happens after that? Nuclein, one more thing is, nuclein is a substance which found in the nucleus. So it is found in nucleus. Okay, nuclein is found in the nucleus. So this is the history. But there was another three scientists, that is Avery, McLeod, and McCarthy. Everyone know that, right? They came and they proved genetic material as in nucleic acid. So nucleic acid is genetic material they proved by their experiment. Everyone knows that, right? Every McLeod and McCarthy, those who have completed their BSc or as well as 12th standard, they know that they proved nucleic acid as a genetic material. Okay, what happened after that? Griffith came, and Griffith done the experiment of transformation. You know that what he did? He took the rats and he inserted S strain, R strain, and after that, with the transformation principle, he proved DNA as a genetic material. That's it. Okay. So he proved the DNA as a genetic material. Now we'll be going to see the functions of nucleic acid. Okay, let's see the functions of nucleic acid. So nucleic acid, we know that it is involved in genetic transformation, and it also acts as carrier molecule, right? Anything. So first point, we can say that it is a hereditary vehicle. Why? Because it is helping the transformation. Okay, so we can call the DNA hereditary material as a DNA as well here, right? DNA is a hereditary material, and they maintain what? They maintains the identity between the generation, maintain identity between two generations. Okay, they can do this one. Another function is, you can say it act as coenzymes, right? It also act as coenzymes, and they act as a source of energy. Obviously, source of energy, it converts ATP to ADP, ADP to ATP, right? So source of energy. And metabolism, it act as a carrier and a carrier molecule in a metabolism. Why? Because they do the phosphorylation reaction, right? So we can say this one as well. And also we can say it is the reserve bank of genes, right? Because it has a genetic information, and called it as reserve bank of genetic information, right? And also we can call them like they are involved in translation, transcription, right? So we can say that DNA directs the synthesis of RNA, and RNA, we can say DNA directs the synthesis of RNA, and RNA directs the synthesis of protein. Here transcription, and here is translation, right? On reverse transcription, it will give the DNA again, and this is a self replication, right? On DNA synthesis, it will give the RNA, and RNA on directed towards the protein synthesis, and that's how the cycle goes on. Okay, so this is the functions of nucleic acid. Now we'll be going to see the structures of purines. Okay, we'll be going to see the structures of purine. Here I'm going to draw the structure. How to draw the structure? First you need to draw like this. So see, I'm drawing the structures. You need to draw it like that. Here you will get a N, nitrogen, okay, attached to it, and here N, this will be attached here, N. So here you can see I have drawn the structures. Before going to explain the structure, I just want to say you that there will be a basis that we call it as nitrogen bases, right? So nitrogen bases are of two types. Nitrogen bases are of two types. We call it as purines and pyrimidines, which are involved in DNA. Okay, they plays a vital role in DNA. So there are nitrogen bases which are of two types, that is purines and pyrimidines. So we are going to see the purines structure, and then we'll be going to see the pyrimidine structure and how they are playing the important role in DNA. We are going to see that as well. So here you can see I have drawn already the purine structures. So purines are of two types. Purines are of two type, only two purines will be there, that is adenine. They are of not two type, they are two, okay, adenine and guanine, that's it. Okay, adenine and guanine. So here the structure which I have drawn is the adenine. So how you are going to keep the numbering for the purine? Purines are always anti-clockwise in direction. You need to write here, they are anti-clockwise in numbering. So you need to give the numbering in an anti-clockwise direction, and you need to understand that purines are derivatives of pyrimidines, because in pyrimidines this ring will not be there, only this ring will be there for pyrimidines. That's why purines are the derivatives of pyrimidines. Okay, now we'll be going to see the numbering. This ring we called it as imidazole ring. What? Imidazole ring, which is present in which amino acid? It is present in histidine amino acids. Okay, histidine amino acids में imidazole ring present होगी. Okay, now let's give the numbering from here. 1, 2, 3, 4. Here you should get a double bond. Okay, and here 5, here 6, then 7, 8, 9. Okay, now you need to give the number, you need to give the name based on this structure. So how you are going to give the structure name? Here at the sixth position there is a amine group, right? So you need to write here 6 amino Purine. That's it. There is no any other functional group present in this string. That's why we are not taking anything. This whole structure you call it as purine, but at the sixth position there is a functional group, that is amine group is there. That's why we are writing as 6 amino purine. Now let's give the name for this structure. Now again we are going to keep the numbering in anti-clockwise direction. 1, 2, 3, 4, 5, 6, 7, 8 and 9. So how we are going to give the name? First we will be going to see the functional groups wherever it is present. So first functional group is present at the sixth position, that is amine, right? And another functional group is present at the sixth position, that is oxy. So we are going to write the name according to that. That is 2 amino, 6 oxy purine. So this is the actual name of the structure, but in that place we will be calling it as a guanine. In another term we called it as guanine, and in another term we called it as 6 amino purine as a adenine. So these are the purines structures and their names. Now we'll be going to see the functions of purines. So if we talk about the functions of purines, there will be very less functions of purines, not less, actually we can say it is more. It is involved in metabolism, and also it controls cell growth, it contributes to sugar transport and donate phosphate group in phosphorylation reaction. So in the next video we'll be going to see the pyrimidine structures, and if you are new on my channel, please subscribe my channel and hit the bell icon so that you can receive more notifications regarding like this. Thanks for watching.